Welcome, curious minds, to another deep dive. Today we're diving deep, really deep, into the fundamental processes that govern all life. Cell growth and cell division.
Mm.
You might think, cells just get bigger and multiply.
Mm.
Simple.
Right. Yeah, that's the basic idea. But as we'll unpack, it's actually far more intricate. Honestly, quite astonishing when you look closely.
Astonishing, I like that. So what are we getting into?
We'll explore the incredible precision, how cells manage their size, their numbers, the really complex signaling pathways involved,
Okay.
and crucially, how disruptions in these processes can lead to serious health issues. Cancer's the big one, obviously.
Right.
And we'll also delve into the sophisticated ways we've learned to intervene, especially with what are known as cytostatic drugs.
Okay, intervention. So our mission today is to kind of demystify these cellular marvels, highlight some surprising facts hidden away in this microscopic world.
Exactly.
And really understand why this knowledge is so crucial. I mean, from embryonic development right through to advanced medical treatments.
It underpins so much.
We've gathered some comprehensive sources to guide us through this biological maze. So let's unpack this. The dance of life. Understanding cell growth and division. Okay, first things first. Let's get our terms straight. What exactly is cell growth? Because it's not just about, like, inflating a balloon, is it?
That's a great way to put it, actually. No, it's not just inflation. Cell growth refers specifically to an increase in the total mass of a cell.
Mass, okay.
Yeah, so encompassing the volume of its cytoplasm, the nucleus, all the organelles inside. It happens when the cell's rate of building biomolecules, that's biosynthesis or anabolism,
Right, the building part.
is greater than its rate of breaking them down, through things like proteasomes, lysosomes, autophagy. That's catabolism.
So it's about accumulating stuff, basically, not just multiplying the number of cells.
Precisely.
And here's where it gets, for me anyway, really interesting. Cell growth is often confused with cell division or the cell cycle, but they're distinct processes. They can happen independently.
Absolutely. It's a super common misconception. Think about early embryonic development.
Okay.
The zygote, the fertilized egg. It undergoes repeated cell divisions, but without much actual cell growth.
Huh.
So the cells just get smaller and smaller, forming structures like the morula, the blastoderm.
Like cutting a cake into tinier slices.
That's exactly. Conversely, think about neurons. Nerve cells.
Yeah.
They can grow significantly, extending those long axons during development, but they don't divide again, or at least don't go through the full cycle.
Oh, wow, okay. So growth without division, that's wild.
It really is.
So in multicellular organisms, tissue growth. It's usually not just cells getting fatter then.
Generally, no. It's usually about cell proliferation. So a mother cell, it grows, and then it divides into two daughter cells.
Right.
And this exponential increase in cell number means, well, 2 cells accumulate mass at twice the rate of 1, 4 cells at 4 times the rate, and so on. That's how tissues can expand so quickly.
Okay, that makes sense. The power of doubling.
It is. And cell size becomes this delicate balance. If growth is disproportionately faster than division, you get larger cells.
Fewer, bigger cells.
Right. And if division increases disproportionately, you get lots of smaller cells. Typically, though, cell proliferation balances these rates to maintain a roughly constant cell size in a given tissue.
But what about those exceptions, those really unusually large cells? How do they work?
Oh, those are fascinating. Some cells get enormous through this weird process called endoreplication.
Endoreplication.
Yeah, instead of dividing after copying their DNA, they just keep replicating their genome over and over. S phase, S phase, S phase, without mitosis or splitting.
So they end up with tons of DNA copies.
Exactly. Highly polyploid. They have many copies of their genome. Oocytes, egg cells are a classic example. They can be huge, especially in species where the embryo develops outside the mother, like in an egg.
Needs lots of supplies.
Totally. They achieve this size either by literally pumping in cytosolic components from adjacent cells via these things called ring canals.
Like tiny pipes.
Kind of, yeah.
Yeah.
Like in fruit flies. Or, as seen in frogs, by internalizing massive nutrient storage granules, like yolk, through endocytosis, basically swallowing supplies. The maestro of the cell. How growth is controlled.
Okay, this control is incredible. It really does sound like there's a biological maestro conducting the whole thing. How do cells manage this mass accumulation?
There absolutely is a maestro, or maybe several working together. Fundamentally, cell growth is driven by increasing the overall rate of cellular biosynthesis, making more stuff.
Right.
This involves expressing genes for RNAs and proteins, including all the enzymes needed to synthesize lipids, carbohydrates, everything.
Yeah.
Gene expression, transcription into mRNA, translation into proteins. It's all regulated by these complex gene networks.
And what's fascinating is how the global rate, the overall volume, can be cranked up. How does that happen?
Well, there are a few key levers. You can enhance the overall rate of transcription itself by RNA polymerase II. Or you can increase the efficiency of translating those mRNA messages into actual proteins.
Yeah.
The latter often involves boosting the number of ribosomes in tRNA, the protein factories, and their delivery trucks.
More factories, more output.
Exactly. For instance, there's a transcription factor called MICUP. It's kind of a master regulator. It can induce the activity of all three major RNA polymerases, driving global transcription and translation, basically hitting the accelerator hard on growth.
And then there's TR. I keep hearing about TR. Seems like a really big player in this.
TR is huge, specifically the TRC1 complex. It's a critical upstream regulator of both translation initiation, starting protein production, and ribosome biogenesis, making more factories.
How does it work?
It's a kinase, an enzyme that adds phosphate tags. It directly phosphorylates and inactivates a protein called 4EBP, which normally put the brakes on translation initiation.
So TR takes the brakes off protein building.
Right. And it also activates another kinase, S6K, which promotes ribosome biogenesis. So less braking, more factory building. It's a central growth promoter.
Okay, so if TR boosts growth, what if you need to slow things down? How do you inhibit growth?
Good question. You can do the opposite. Decrease the global rate of gene expression. Or you can increase the rate of breaking things down, enhancing autophagy, the cell's recycling system.
Ah, autophagy.
Yeah. And interestingly, TUR normally inhibits the main autophagy-inducing kinase at G1. So reducing TUR activity does two things. It lowers translation, and it increases autophagy. Slows construction, speeds up demolition, reduces growth overall.
A double whammy.
Pretty much. Which raises the big question, how is all this regulated in whole animals?
Right, because individual cells don't just decide on their own. There must be signals.
Growth factors.
Exactly. Many of the signal molecules controlling animal cell growth are called growth factors.
And what pathway do they typically use? Is it related to TUR?
Often, yes. Many induce signal transduction via the PI3 KLA-KTM-TOR pathway. It's a cascade.
A chain reaction inside the cell. Precisely. It involves a lipid kinase, PI3K, and a protein kinase act upstream, which then activate TTOR. TOR, in turn, promotes translation and inhibits autophagy, driving cell growth.
And nutrients are key here too, right? You can't grow without building blocks.
Absolutely fundamental. Nutrient availability directly influences the production of growth factors, like those in the insulin IGF-1 family.
Ah, insulin.
Yeah, these circulate as hormones in animals, activating that PI3K-TM-TOR pathway, promoting TOR activity. So well-fed animals, lots of nutrients, they grow rapidly.
Makes sense.
While animals lacking sufficient nutrients dial down their growth rate. And fascinatingly, recent work shows even cellular bicarbonate metabolism, essential for making building blocks, can be regulated by MTORC1 signaling. It connects everything.
So, pulling this together, what does this mean for a cell's decision to divide based on its size? Is there a size checkpoint?
It's still a bit debated, maybe controversial, but there's a theory that many mammalian cells do undergo size-dependent transitions in the cell cycle, controlled by that key division enzyme, CDK1. Now, we know the proteins that control CDK1, but their precise connection to how a cell monitors its size, that remains elusive. One model suggests mass is the driver.
The actual weight of the cell.
Sort of, yeah. The idea is a cell initiates S-phase DNA replication, leading eventually to division only when it hits a certain size or mass. And conversely, later cell cycle events are delayed until the cell mass is sufficient for S-phase to begin. It prevents cells from getting progressively smaller.
Regulating cell dimensions.
Speaking of size, it varies wildly, doesn't it? Tiny bacteria, you mentioned, single-celled algae meters long. How do they decide how big to get before splitting? It seems like such a fundamental question.
It is. It's one of the hottest, most fascinating areas in cell biology. How does a cell know its own size? Chemical gradients are part of the answer.
Okay.
And there's a hypothesis that mechanical stress detection, maybe through the cytoskeleton, the internal scaffolding, is involved too. But a lot of research focuses on simpler organisms where the cell cycle is really well understood, like yeast.
Yes, yeast, the biologist's friend, especially those wee mutants you mentioned earlier.
Exactly. The relationship between size and division has been studied intensely in yeast. We know for some cells, division doesn't start until a certain size is reached.
Right.
If you restrict nutrients, slowing down growth, The time between divisions actually gets longer. They wait.
They wait until they're big enough.
Correct. And those Wee mutants, which start dividing before reaching normal size, they were absolutely instrumental in figuring this out.
And that led to the discovery of the Wee1 protein, the break.
It did. Wee1 is a tyrosine kinase, an enzyme that inhibits CDC2, which is the human CDK1 equivalent, the engine for mitosis. Wee1 phosphorylates it, puts a tag on it.
Keeps it inactive?
Exactly. Keeps it inactive during the early G2 phase when cells are still small. Then, when cells reach sufficient size, another enzyme, a phosphatase called CDC25, removes that inhibitory tag.
Activates CDC.
And lets the cell enter mitosis, boom.
So if Wee1 activity is weak, cells divide smaller. Makes sense? Suggests maybe Wee1 gets diluted as cells grow. But you said it's more complex, something about location.
Yeah, it gets really intricate. What's fascinating is the connection between other protein kinases, CDR2 and CDR1, which actually inhibits Wee1.
Okay, an inhibitor of the inhibitor.
Right. And their specific location in the cell, they gather in a band of cortical nodes right in the cell's middle during interphase, the growth period.
Nodes, like little hubs.
Exactly. And after mitosis, cytokinesis factors like myosin II that helps pinch the cell in two, they also go to similar nodes, which then condense into the cytokinetic ring. There's even another protein, BLT1, that hangs out with CDR2 there. Knock out BLT1, cells get longer, mitosis is delayed.
So these physical spots, these medial nodes, are crucial control centers.
It seems so. CDR2 seems to be at the top of this hierarchy. It orchestrates the assembly of these nodes.
Okay.
And it promotes mitosis by negatively regulating Wee1. But here's the twist. CDR2 also recruits Wee1 to these medial cortical nodes.
Wait, it brings the break to the spot where the anti-break signals are.
Exactly. It localizes Wee1 right there with its own inhibitory network. So mitosis is controlled through CDR2-dependent negative regulation of Wee1 at these specific sites. It's incredibly spatially controlled.
That's mind-blowing precision. How do cell polarity factors fit in, keeping things in the right place?
They provide the spatial cues. Cell polarity factors at the cell tips essentially tell CDR2 where not to be, limiting its distribution to the cell middle.
Ah, okay.
In fish and yeast, S. pombe, cells divide at a very defined size because CDK1 activity is tightly regulated. There's a polarity protein kinase called POM1 that localizes just to the cell ends.
Right at the tips?
Uh-huh. Knock out POM1, CDR2 isn't restricted anymore, it spreads out, and the cells divide at a smaller size premature mitosis.
So POM1 is like a fence, keeping CDR2 corralled in the middle.
That's a good analogy. It provides inhibitory signals that confine CDR2.
So POM1 could be acting as a size sensor then. How would that work?
Well, think about it this way. POM1 forms gradients, peaking at the cell ends. As the cell grows longer, the distance from the ends to the middle increases.
Right.
So the POM1 gradient, its influence, diminishes in the middle as the cell elongates. In small, early G2 cells, the POM1 signal in the middle is strong enough to keep CDR2 inactive, preventing mitosis.
Okay.
Only as the cell grows into late G2, when POM1 is more confined at the tips and its signal weakens in the middle, is CDR2 in those medial nodes activated.
And then it can inhibit Wee1.
Exactly, allowing mitosis to proceed. It provides a direct link between cell size, the physical length, and the biochemical regulation of mitosis entry, all through this POM1, CDR2, CDR1, Wee1, CDK1 pathway.
Wow, that is elegant. What about other organisms? You mentioned cell fusion.
Yeah, that's another way to get big cells, forming syncytia. Skeletal muscle cells are the classic example. Thousands of individual precursor cells, myocytes, fuse together to form these huge multinucleated fibers, inches long sometimes.
Incredible.
And genetic studies, especially in fruit flies, have identified the specific genes needed for that myoblast fusion process. In plants, of course, it's different again because of their rigid cell walls.
Right, but walls complicate things.
They do. Growth involves carefully remodeling those walls, often under the influence of plant hormones, to allow the cell inside to expand.
And even bacteria have clever tricks.
Oh, yeah. Rod-shaped bacteria like E. coli, Colobacter, B. subtilis. They seem to control size by adding a constant volume between divisions.
A constant amount each time.
Yeah. So if a cell happens to be born a bit smaller, it adds that constant amount and gets closer to the average. If it's born bigger, adding the same amount makes it proportionally less oversized. Over generations, they naturally converge to an average size. It's quite neat, the cell cycle, division in detail.
Okay, so we've covered growth, size control, incredibly complex. Let's talk about the actual division now. The cell cycle, the splitting process itself.
Right, so for most cell components, growth is pretty continuous. except for a brief pause during M phase. That's when the nucleus divides, and then the whole cell splits in two.
M phase, mitosis.
Mitosis and cytokinesis, yeah. The entire process, growth and division, is the cell cycle, and it has four main phases.
Can you walk us through those quickly?
Sure. First is G1 phase, that's gap 1. The cell is growing, synthesizing enzymes needed for the next step.
Which is?
S phase, synthesis, that's when DNA replication happens. You go from one copy of each chromosome to two identical copies, the sister chromatids.
Okay. G1, then S.
Then G2 phase, gap 2. More growth, significant protein synthesis, especially making microtubules needed for mitosis, getting ready for the big event.
And finally M phase.
Finally M phase, that's nuclear division, karyokinesis, followed by cytoplasmic division, cytokinesis forming a new cell membrane to separate the two daughters. And M phase itself has distinct stages: prophase, prometaphase, metaphase, anaphase, telophase, leading up to cytokinesis.
A choreographed dance.
Very much so.
So what are the different types of division? You mentioned mitosis and meiosis earlier.
Right. Cell division is simpler in prokaryotes, bacteria. They use binary fission, basically. Replicate DNA, segregate the copies, split the cell, done.
Straightforward.
Eukaryotic cell division, in contrast, involves either mitosis or meiosis. Now, both binary fission and mitosis produce two daughter cells that have the same number of chromosomes as the parent cell. Identical copies, essentially.
Okay.
Meiosis is different. It's more complex, specific to diploid organisms, those with two sets of chromosomes, like us. It produces four special daughter cells called gametes. Sperm or eggs.
And they have different chromosome numbers.
Yes. Each gamete has half the normal amount of DNA, half the number of chromosomes.
So our body cells have, what, 46 chromosomes?
Exactly. Diploid, 2 and 46.
But our sperm and egg cells have only 23.
Precisely. Haploid, N23. Let's track it. Before DNA replication, a cell has Z chromosomes. After S phase, it has two Z DNA, but still technically Z chromosomes, each made of two chromatids. In mitosis, that 2Z DNA is divided equally, so each daughter gets Z chromosomes, same as the parent. In meiosis, there are two division steps.
2 rounds of splitting.
Yeah, meiosis 7 and meiosis the second. The result is 4 daughter cells, each with N chromosomes half the original number.
This brings up a really fundamental question. Why bother with meiosis? Why sexual reproduction? Mitosis makes perfect copies, seems way simpler. Why the complexity?
That's a huge question in evolutionary biology. You're right, mitosis is great for making identical copies. But sexual reproduction, enabled by meiosis, gives a massive selective advantage.
How so?
During meiosis, specifically in meiosis, I have homologous chromosomes, the pair you got from each parent, say chromosome 7 from mom and chromosome 7 from dad, they physically pair up.
Okay.
And they can interact and exchange segments, genetic recombination or crossing over. Information swaps between them.
Shuffling the genetic deck.
Exactly. In mitosis, these homologous chromosomes don't interact like that. This recombination is, 1, a fantastic mechanism for repairing DNA damage, but 2, crucially, it creates new combinations of genes, new alleles on the same chromosome.
Generating diversity.
Precisely. Some of these new combinations might be beneficial, helping the offspring adapt better. It fuels evolution. Plus, in organisms with multiple chromosome sets, sex also allows for different combinations of alleles homozygotes and heterozygotes according to predictable ratios, which also influences population genetics. It's all about variation. When growth goes wrong, disorders, and their control.
Okay, so this incredibly precise control of growth and division is clearly vital. It has to be, right? What happens when it breaks down?
Well, when it goes wrong, you get a range of growth disorders at the cellular level, and these fundamentally underpin much of what happens in cancer.
Right. Cancer is uncontrolled growth.
Uncontrolled growth and division, yeah. Beyond normal limits. Plus, often invasion into nearby tissues, and sometimes metastasis spreading to distant sites via lymph or blood.
So disruptions in cell growth itself are a key feature.
Absolutely. Key determinants of cell growth, like ploidy having the wrong number of chromosome sets.
Which you mentioned earlier.
Right. And the regulation of cellular metabolism. These are commonly messed up in tumors. That's why heterogeneous cell growth, or pleomorphism cells, having varied sizes and shapes, is one of the earliest visual hallmarks pathologists look for when diagnosing cancer progression.
Different size bricks in the wall.
Exactly. And while its role in disease progression isn't always fully clear, we know that in epithelial tissues like linings, misregulation of cell size can cause packing defects. Things don't fit together right. And this might actually help aberrant cells disperse.
Given how vital understanding all this is, how do scientists actually measure cell growth? What are the tools?
Good question. There are quite a few methods. For just looking at cell size growth, microscopy with suitable stains works. You can see them getting bigger. But often, the increase in cell number is more critical. That can be done by manual counting under a microscope, using techniques like the dye exclusion method with trypan blue to make sure you only count live, viable cells.
Sounds tedious. Are there faster ways?
Oh, definitely. Less fastidious, more scalable methods use cytometers. Flow cytometry is particularly powerful.
Flow cytometry.
Yeah, it shoots cells single file through a laser beam. It can count thousands of cells per second and measure other parameters simultaneously using fluorescent probes.
Like what?
You can distinguish dead versus viable cells, different cell types in a mix, track cell differentiation, or even quantify the expression of a specific biomarker like Ki-67, which indicates proliferation.
Very cool. Could you actually measure the mass of a single cell, like weighing it?
You actually can. It's amazing. The total mass, including water, can be measured in real time over hours using something called an inertial picobalance.
Picobalance. Tiny scales.
Incredibly tiny. Or you can measure a cell's buoyant mass, its total mass minus the fluid it displaces, using suspended microchannel resonators. Gives you an idea of density changes too.
Wow. And what about metabolic activity? Is the cell active?
Yep, another angle. Metabolic activity growth can be assessed using assays. Things like CFDA and calcin-AM are fluorometric. They measure membrane integrity and enzyme activity.
Okay.
Then there are MTT assays, which are colorimetric, or Resagerin assays, which are fluorometric again. These often gauge mitochondrial redox potential, basically, how active the cell's powerhouses are.
Do these different methods always agree?
Not necessarily. That's important. They measure different things: membrane function, enzyme activity, mitochondrial power. They might not correlate perfectly depending on the cell conditions or what you're treating them with. It gets complex, especially if you have mixed populations or drugs that might be toxic and affect growth differently.
The science of cytostasis.
Okay, so we've looked at growth, division, measurement, what goes wrong. But what about the opposite? Stopping cell growth?
Okay.
Intentionally.
Right. That brings us to cytostasis. The word itself tells you. Cyto for cell and stasis for stoppage.
Cell stoppage.
Exactly. The inhibition of cell growth and multiplication. So a cytostatic substance refers to something, a cellular component, or more commonly, a medicine that inhibits cell division. It can sometimes even induce cell death, though that's technically cytotoxicity.
And why is cytostasis important? Obviously for treating disease, but is there a bigger biological picture?
Absolutely. If you connect this back, cytostasis, regulated inhibition, is a fundamental prerequisite for structured multicellular life.
How so?
Without finely tuned mechanisms to stop cells from growing and dividing at the right time and place, we wouldn't have tissues or organs. We'd just be, well, unorganized heaps of cells. Like a tumor, essentially.
So brakes are just as important as accelerators.
Precisely.
Medically, where do we see cytostatic drugs used most?
Chemotherapy for cancer is the biggest one, by far. But they're also used in treatments for some skin diseases like severe psoriasis and even certain infection treatments. But it's crucial to remember they're generally nonspecific.
Meaning they hit healthy cells too.
Yeah, especially other rapidly dividing cells. That's why you get side effects. We'll come back to that.
Okay. Are there natural things in the body that cause cytostasis?
There are. Nitric oxide, NO, is a good example. Activated immune cells, macrophages, can produce loads of NO.
Nitric oxide, like laughing gas.
Related molecule, but different context. This NO, produced by immune cells, induces both cytostasis and cytotoxicity in tumor cells. Been shown in vitro and in vivo.
How does it stop growth?
One key target is ribonucleotide reductase, an enzyme essential for making DNA building blocks. No inhibits it, usually reversibly, though other targets probably contribute too.
Any other natural activators?
Well, things that activate those macrophages can indirectly cause cytostasis. Lipopolysaccharide, LPS, a bacterial component, and LAP, lipid A-associated protein, are potent macrophage activators that stimulate tumor-killing activity, including cytostasis.
Interesting. What about diet components, fats?
Yes, certain polyunsaturated fatty acids, PUFAs, specifically N3s and N6s, show distinct effects. Cystostatic concentrations of these PUFAs, in some studies, didn't immediately kill cells, but caused permanent growth arrest by messing with the cell cycle.
How?
Metabolites from the lipoxygenase pathway seem to be involved.
Yeah.
But importantly, like many natural effectors, this PUFA cytostatic activity isn't necessarily tumor-specific.
Okay. Can you give us some concrete medical examples where cytostasis has been seen in action from the sources?
Sure. In breast cancer research, nitric oxide showed a cytostatic effect on a common human breast cancer cell line. MDA, MB231.
Slowed it down.
Exactly. And the study also noted it could induce apoptosis, cell death after longer exposure, about 48 hours, and those long-chain PUFAs we mentioned. They've been shown in vitro to inhibit division, cause cycle arrest, and induce death in malignant epithelial cells from various tissues.
The arsenal against cancer?
So, cytostatics,
Yeah.
they're basically chemotherapy drugs, right?
Yeah.
So, how do they actually work to fight cancer? What's the strategy?
That's right. They're the classic chemotherapy agents. They're designed specifically to inhibit or stop the growth of cancer cells
How?
by interfering with the cell cycle in various ways, disrupting DNA replication, messing with RNA transcription, blocking the machinery of mitosis, hitting critical processes needed for cell division.
But you said they're nonspecific.
Correct. Unlike more modern targeted therapies, cytostatics generally hit any rapidly dividing cell. That includes cancer cells, yes, but also healthy cells in places like hair follicles, the lining of the gut, and bone marrow where blood cells are made.
Which explains the side effects. What are the different types, the categories of these drugs? How do they differ?
There are several main classes, each with a different mechanism of action. First, alkylating agents. Things like cyclophosphamide, ifosfamide. They add alkyl groups, chemical tags to DNA.
Like sticking gum on the DNA?
Sort of. It causes DNA strands to cross-link or break, leading to cell death. Then, antimetabolites. Methotrexate, 5-fluorosyl-5-FU, cytarabine. These mimic natural building blocks needed for DNA or RNA synthesis.
Big parts.
Exactly. They get incorporated or block enzymes, messing up replication. Anti-tumor antibiotics, like doxorubicin, blumycin. These bind to DNA, causing breaks or preventing it from unwinding for replication. Plant alkaloids, also called mitotic inhibitors, vincristine, paclitaxel, etoposide, these disrupt microtubules.
The cell's internal skeleton.
Yeah, the dynamic scaffolding essential for separating chromosomes during mitosis, no scaffold, no proper division. And topoisomerase inhibitors, irinotecan, topotecan. Topoisomerases normally help untangle DNA during replication. These drugs block them.
Leading to DNA knots and breaks.
Big time, causing lethal DNA damage.
Quite an arsenal. How are these drugs usually given to patients?
The most common route is intravenously, IV infusion, directly into the bloodstream.
Okay.
But other methods are used too. Oral tablets or capsules, injections into muscle or under the skin, intramuscular or subcutaneous. And for some cancers, like leukemia involving the central nervous system, intrathecal administration, injecting directly into the cerebrospinal fluid.
Right into the spinal fluid.
Yeah, to bypass the blood-brain barrier and hit cancer cells hiding there.
Given that nonspecific action, what are the typical side effects people experience? We touched on it. But
It's the consequence of hitting all fast-dividing cells. So hair loss, alopecia, is common because hair follicle cells divide rapidly.
right.
Nausea and vomiting because the gut lining cells turn over quickly. Fatigue is very common. Low blood counts are a major issue. Anemia from low red cells. Leukopenia from low white cells leading to infection risk. Thrombocytopenia from low platelets causing bleeding risk.
Sounds rough.
It can be. Also, mouth sores, diarrhea or constipation, and potential fertility problems. However, and this is important, supportive care has improved dramatically. We have much better anti-nausea drugs, antiemetics, growth factors to boost blood cell recovery, steroids. These help manage and reduce side effects significantly compared to decades ago.
That's good to hear. Do doctors usually use just one of these drugs or combine them?
Very often they're used in combination. It's called polychemotherapy.
Why combine them?
Several reasons. It increases overall effectiveness, hitting the cancer with multiple weapons. Different drugs might target cells at different stages of the cell cycle. And crucially, it reduces the risk of the cancer developing resistance to a single drug.
Makes sense. Attack from multiple angles.
Exactly. You see standard combinations like CHO for lymphoma or FOLX for colorectal cancer. Many standardized regimens exist.
Well, what about the bottom line? How effective are they? Are there success stories?
Absolutely. Despite the side effects and the development of newer therapies, cytostatics remain a cornerstone of cancer treatment. They've dramatically improved survival rates. Many cancers that were once fatal are now highly treatable or even curable.
Like which ones?
Childhood leukemia now has cure rates over 80%. Testicular cancer, thanks largely to cisplatin-based chemotherapy, is over 95% curable. Hodgkin's lymphoma has very high remission rates, and adjuvant chemotherapy given after surgery has significantly boosted survival for common cancers like breast and colorectal cancer. It's been transformative.
That is incredible progress. Are there new developments? Are we still improving these older drugs?
Oh, definitely.
Yeah.
Research continues. One area is dose-dense therapy, giving treatments closer together, sometimes with growth factor support, to increase the intensity and impact.
Hitting harder, faster.
Kind of. Then there's the push towards personalized medicine, trying to match specific drugs to the genetic profile of an individual's tumor, although this is often more developed for targeted therapies than traditional cytostatics, and better drug delivery systems. Things like liposomal formulations packaging the drug inside tiny fat bubbles. The idea is to help deliver the drug more specifically to the tumor, potentially reducing toxicity to healthy tissues.
Interesting. One last thing on this. Thinking bigger picture again. What happens to these powerful drugs after they've done their job in the patient? Where do they go?
That's a really important point and an area of growing concern. After administration, cytostatic agents and their metabolites are expelled from the body, mainly in urine and feces.
So they end up in wastewater.
Exactly. They eventually reach wastewater treatment plants and, potentially, the wider aquatic environment. This poses a potential ecological risk. While some studies exist, we definitely need more research to fully understand the environmental impact and how to mitigate it. Outro.
Wow. Okay. That was quite the journey. A really deep dive into this.
It really is fundamental, isn't it? From the intricate dance of molecules dictating a cell's mass, that precise timing of division, it's governed by these incredibly sophisticated mechanisms.
Almost unbelievable complexity packed into something so small.
And when those mechanisms go wrong, especially in cancer, we see the devastating impact. But on the flip side, the development and refinement of cytostatic drugs, of chemotherapy, it really represents a remarkable triumph of ingenuity, harnessing biology to fight biology.
Saving countless lives in the process.
Absolutely.
It's just a powerful reminder, isn't it, that understanding the smallest building blocks of life can have the absolute biggest impact on our health, our future. So here's something to think about. We've talked about how cells sense their size, how they follow these intricate internal programs. If a single cell exhibits such complex, almost intelligent self-regulation, what does that suggest about the nature of biological organization itself at every scale? Just something to ponder.